Good morning, students. Today, let's go for Operon concept. To know the Operon, we'll have to understand that in bacteria, mainly in bacteria, number of genes unite together to carry out a function. And that generally we find in case of bacteria. What are those? We'll be trying to know. See, in that case, you are finding this is a gene which is having some part. This is I, which is called regulator. Then this is P, promoter. O, this is called operator. Okay? Then this part, it is called structural gene. What is called? structural, structural genes. And these structural genes, dear students, has three most important genes. Here, Z, Y, A. Now we're trying to understand the metabolism of lactose. So, this operon that we are going to study is called Operon. Lac means from lactose. What happens exactly? You try to get it. The regulator is producing repressor. Repressor mRNA. Now what is that mRNA? We're trying to bring the information that we have gathered in case of the protein synthesis, transcription. So the gene will be transcribing mRNA, and that is called the repressor mRNA, which produces repressor. Got it? Let me keep it up to here. What is the function of promoter? Promoter prompts. It gives the information to the operator. Then the RNA polymerase helps in the transcribing the mRNA, which lead ultimately to the formation of enzyme. Z, structural gene Z, produces beta galactosidase. Y, it produces permease. And A, it produces trans-acetylase. Okay? So Z is producing what? Beta galactosidase. Y is producing what? Permease. And A, structural gene A, produces trans-acetylase. Now try to get it. How it happens? It has happened due to one enzyme. I'm writing down the name of the enzyme here. RNA polymerase. This RNA polymerase helps in transcribing. See, first thing, two things. In protein synthesis, we have done transcription, then translation, isn't it? So, this RNA polymerase will help in the transcribing the mRNA. Over. Now, that mRNA that is produced will be producing enzyme. What is enzyme? We have done in class 11. All enzymes are proteins. Definitely we are going for translation. So Z, structural gene Z, will be producing beta galactosidase. Y will produce a permease, and A will be producing trans-acetylase. So let me come to this point. Now repressor, when it binds with the operator, understood the thing? Then in that case, The promoter gives the information to the operator. And operator, what it does? It tells RNA polymerase not to transcribe the mRNA, not to allow these genes to transcribe. So, what happens? It is in switch off condition. It is called switch off condition. Why? Because the structural genes doesn't produce the transcribed mRNA or lead to the formation of enzyme. Now we are coming to the another thing. Inducer. Another thing is inducer. Inducer in this case is lactose. So what this lactose does? It unites with the repressor and it forms a complex. Understood? Lactose unites with the repressor and forms inducer repressor complex. So what it does? It doesn't allow the repressor to bind with that of the operator. When it is not binded, then it comes to switch on condition. What it is now called? Switch on condition. And in that case, what happens? Operator gives the direction for the RNA polymerase to transcribe mRNA. First mRNA transcription will be there. Then it produces enzyme. So you are finding two things now. One is the switch on, other is the switch off. Just as we found in case of protein synthesis, initiating codon AUG and terminating codon in case of protein synthesis translation. What are they? UAA, UAG, UGA. Here also you are finding the same thing. Switch off when the repressor will be binded with the operator. Switch on when the lactose doesn't allow the repressor to be bonded or to get binded with the operator. Then that is the formation of enzyme. That's what we find in case of the operon. Though it is in bacteria but it is very important in knowing the concept for the metabolism of the lactose. Thank you, students.